Hi everybody, my name's Pat from Field of Mars EEC. Soon I'll be seeing you guys on your field study excursion. Today I've got a couple of guests here who are visiting us in the bushland, and this is Meredith and this is Anne from
G'day.
Applied Ecology. So thanks for coming in, guys, and tell me what line of business are you guys in?
We do a lot of flora and fauna surveys in New South Wales, lots of work in the Sydney Basin, but out west as well.
Gotcha. And who do you guys work for?
Mainly councils that are the managers of the local reserves in many cases. So yeah, mainly councils that we work for.
Okay, so that's who hires you to prepare those surveys and reports, but is there a company that you belong to, and who's the boss of that company?
Yep,
Yes.
I'm the boss.
No, I'm the boss. Applied Ecology, and we co-own it, and then we employ ourselves and some other people. I do a lot of flora and vegetation surveys. I've got a doctorate, and that's in ecohydrology.
And my specialty is fauna. I have always loved fauna. I grew up on the Central Coast and I volunteered at the Australian Reptile Park from about the age of 11. So I've kind of been involved with fauna since then. Fell in love with reptiles, and that grew to a love of all fauna obviously as I got into the industry.
Well, I was always nicking off from school and going down bush. It was somewhere to go, but I loved being there. Do something you love.
Yes, exactly. And I've always loved animals. Like I was a member of what was the Wires equivalent back then, and sort of was, had joeys by the time I was in year 11 in my bedroom, and sugar gliders also in my bedroom, and all that kind of stuff. So, and it just grew from there. And then I worked for national parks, and here I am now, a degree and a master's later in wildlife management. So. Well, there's the simple things as the preparation for being outdoors. You've got to be fairly physically fit, and in a laboratory it's very controlled. Not only the experiments that you're running, but the environment that you're in, where outside weather, people, other events can interrupt your field work. So you have to be very flexible when you're sort of working in the field. On a large scale, it's the season. For fauna, it's particularly important to try and sample in different seasons. You'll get your highest levels of activity in spring and then in autumn. In summer it's often very hot, and in winter it's cold, and the migratory species leave in winter. So if you want to do a really good fauna survey where you're trying to detect the maximum amount of species, spring is the way to go. And then you've got to work around the weather within the spring. The spring this year has been very, very dry, and now we're expecting some wet weather as well. So we'll have to redo some of the surveys we did when it was dry once it rains, and that will help us detect the most species that we possibly can in the 3 months of spring.
It'd be much the same for the flora. So if you're not really familiar with the flora of the area, it's much easier to identify a plant if it's got flowers on it. Threatened species, particularly before heading out in the field, it's really important that you check when they flower, because there's no point in looking for something that you'll only find if it's flowering, if that's not its flowering period. Yes is the short answer. It could be something as simple as, is the mapped vegetation community there? And that's confirmed by the suite of species that are present. Or it might be, as an example, the project we've been working on here, we're looking at replicating some surveys that were done 10 years ago, and we're looking to see changes over time, and then what might be causing these changes.
And things that often the land managers are interested in are species that are regionally significant, like the swamp wallaby. They want to know what habitats could they possibly be using in these remnant reserves. So we try and investigate which habitats these animals are using. And you can go to the literature and read a lot about it, but if an animal's living within a remnant, you've got to base your answer on that remnant, because that can be very different to how they're living in areas of much more contiguous bush. It's always a good idea to have a base level of knowledge around what animals you might come into contact with, so that then you can familiarise yourself with their calls and what they look like. So you can go to field guides, which are really helpful. They've got maps, distribution maps, and also there's a wildlife atlas or BioNet that you can access online. For birds, there's also eBird, which you can access online, and that'll have records that people have seen in a particular reserve. So you could look up Field of Mars and find out what other birds people have seen here. And that's always a really useful starting point.
So Anne, what sort of methods and equipment do you use for surveying fauna?
Yeah, well, there's different techniques for different groups of species. So for birds, a lot of it is listening, using binoculars, taking photos. You can record them remotely as well, but and there's a song meter that does that, which we've got out in the field at the moment. But normally, it's just binoculars and listening. Half of bird watching is listening for the calls. For mammals, there's a variety of techniques. Most mammals are nocturnal, and you can spotlight for them, and you'll pick up a certain amount by spotlighting. But some of the more cryptic species, you might have difficulty detecting them like that. So we use a technique of hair tubing, and this is a hair tube. And in the back here, you put in some bait, which is peanut butter, rolled oats, and honey. And then that's either placed on the ground if you're targeting things like bandicoots or antechinus, or up in the tree if you're after sugar gliders and other possums like ringtails and brushtails. So when you're surveying an area such as this, you're fairly confident that you're going to see brushtails and ringtails. So with this sort of equipment, you're really targeting the species that are a little bit harder to find. So potentially like sugar gliders and antechinus and things like that.
Do you mind if I hold that up to the camera?
Sure.
So the guys can get a close look. So that's where your little mammal goes in, and the bait's at the back here, and I'm not sure how to open that, but I suppose if you've got a degree in this, it all works out. Oh, there we go. So it all fits in there. So there's little sticky two-sided tape on the inside here,
right.
and that's what the animal's hairs get lodged on, and they're sent off for analysis and identification. Is that right?
They are, yeah. You leave them out sort of like for 5 or 10 days, and you come back and you collect the tape off them, and you just keep it, you know, potentially in a book like this with some like baking paper on top so it doesn't stick to everything.
Bring it up a bit higher. I'm not sure it's in the frame.
Yep, yeah. And that's 2 pieces of tape I collected this morning. I'm hoping there's some sugar glider fur on that, and that'll be sent off to the lab tomorrow. So. Cool. And they take a cross section of the hair, and it's almost like a fingerprint actually, so they can get it down to species level pretty easily.
So, and what I like about that is it's not just the equipment and the methodology, but you're really taking care of the handling of the data. Yes. You've got a special notebook, and you've got the baking paper to make sure there's no cross-contamination between the samples or anything like that.
That's right. And when we put them out, we take a waypoint with the GPS, and I always note which hair tube they came from, so then we can map up where different species were detected via this particular method. This is a bait box that we use in conjunction with a camera, a remote camera, which is triggered by movement. That's been out for about 10 days with this particular camera, looking for sugar gliders again in the field of Mars. So the bait is still in reasonable condition after 10 days. You can see it there. It's just peanut butter, rolled oats and honey. I'll leave that there for a bandicoot to find. So we hang that up in a tree and point the camera at it essentially, or on the ground, depending on which species you're targeting. Let's have another look at the camera.
That's a piece of work, isn't it?
Yeah. Goes up that way.
Yep. Oh, you've got a little solar thing there.
Yeah. This one's a solar charged one, so we can leave this one out for a very long time. This one's been out for 10 days. It's got over 15,000 photos on it, which I will process tomorrow, and hopefully there'll be a sugar glider on it.
So when you say process, does that mean you're going to look at 15,000 photos?
That means I'm going to look at 15,000 photos.
Right, so, okay. Have fun tomorrow.
That's right.
Yeah. And what's this little thing here?
This is an Elliott trap. We use those for small mammals. They come in all different sizes. You can see it pops up like that. It folds down so that you can carry it more easily into the bush, because you normally put out about 20 of these at a time at a particular site. So somewhere like the Field of Mars, we'd put 20 out near Strangers Creek, 20 out near the cemetery, like maybe 20 out on Buffalo Creek, and maybe 20 out around the Environment Centre down here. It's kind of the same technique. Is you put the same bait, the peanut butter, rolled oats and honey in the back, and there's a little pressure plate, so when the animal walks in to investigate the bait, it closes the little door, and the animal is trapped overnight. We put some nesting material in there so they don't get cold, and if there's likely to be rain, we certainly bag them up in a plastic bag so that it's completely waterproof.
So I think you just answered the question that popped into my head, but is there anything special for the animal ethics in handling vertebrates, and I think you just sort of mentioned that sort of stuff
Yeah,
there.
there is. You do have to have ethics approval to do any sort of trapping in New South Wales, and there's a whole lot of protocols which are guidelines around the handling and treatment of animals, and we certainly adhere to those and even go the extra mile really. I mean, I quite like checking the traps at night and letting the animals go. The other thing I try to avoid is like trapping when there might be lactating females. You don't want to trap a mother and keep her in the trap overnight when she's got babies somewhere, or a pregnant female. So I don't do a lot of trapping in spring. This stuff's probably pretty obvious, but you need a fairly good camera to try and capture pictures of animals. And even if you don't get a perfect photo, it's good just to have it for a record for your own confirmation. If you're not sure of a species, you can get a photo and then have a look at it later. I also have a microphone on mine, which is great for just recording frogs and bird calls that you might not be sure about. And you do have to be careful with spotlighting, because we don't like having light shine in our eyes, and neither do the animals, so it's really just a quick look, there it is, and put it away. A big part of the night work is listening as well, just not having any lights on at all and listening, because most animals have very distinctive calls.
So, I mean, you guys have been doing this for a job for a long time, so presumably you're sort of pretty dialed in with your own sensors, and it may be perhaps that you bring back more equipment on your second visit based on what you see or hear, or the scats, I guess, that you see on the first trip.
Yeah, absolutely. An example would be we detected a sugar glider via the hair tubes after, probably 4 months of having cameras out and hair tubes. So I'm targeting that particular area with more hair tubes and putting more remote cameras set up quite high in trees, trying to get some vision of them. It's always good to confirm via more than one technique if you can. Bat fauna, because we don't see them and we don't hear them, we see them very occasionally, they can kind of be forgotten, but they are a big special part of the fauna of the Sydney Basin. And this is a bat detector, which is essentially a recording device, because there's only 2 species of bats that the human ear can hear. They emit a sound at a much higher frequency than what we can hear, and this records it. And then you can look at it visually on a computer screen when you get home, and the shape of the call and how long it goes for.
So you can see the wavelengths and
That's right.
the pitch and the pattern of the call, and that helps with identification.
Absolutely.
It's cool.
Yeah, it is cool. Yeah. And the way that came about is people would catch bats, let them go and then record them. So you knew what the species was in your hand, you let it go, and you continue to record it, so you end up with a library of calls of that species make.
So, Anne, continuing along with survey techniques, here's a biotic factor, which is the surveying of scats, animal poo and other sort of things like that. What have you got here?
Yeah, well, I've got some bandicoot scat and some fox scat. This is just 2 of the sort of scats that you can find pretty readily in the reserves around here. They're very useful and again, we send them off to the lab. So with foxes, you can have a look at what the foxes are actually eating. So they can have a look at what sort of hair and bone is in the fox scat, so that you get an idea of what they're actually preying on in your local area. The same with owls, because owls regurgitate the feathers and fur and bones of animals that they've consumed. And those pellets can be sent off, and they give you a very good idea of what owls have been eating, whether it's brushtails, sugar gliders, ringtail possums, black rats, so you get an idea of what they're into in a particular season. The other thing that it's useful doing scat surveys is you get an idea of what habitats the animals are using. It's a major technique for like detecting koalas, where you mightn't see the koalas themselves, but you can certainly readily find the scats under their feed trees. So quite often you can find the scats and not the animal. It's the same with animals like swamp wallabies in these sorts of reserves. It's easier to find the scats than it is to find the animal. And finding concentrations of scats gives you an idea of where they might be taking refuge during the day. So they're very, very useful. You've just got to remember that if you're handling fox scats at all, that you should use gloves, put them in a brown paper bag, and because they do carry some nasties, but the other ones, not so much.
You talked about the refuges used by swamp wallabies. What would that look like, a swamp wallaby refuge area?
They like very, very dense vegetation, and they basically hide from predators. And not that they have a lot of predators per se anymore in these reserves, but they don't like being disturbed. If they are disturbed by humans or dogs, they will flush out of their refuge areas and run. And we certainly don't want that to happen to them because they could end up anywhere. Bandicoots are very similar. They have very dense refuges during the day. They don't dig burrows as such. I think a lot of people think they do, but they don't. So they also seek very dense vegetation. In some of the remnant reserves, that is often an area of dense weeds, which are sort of impenetrable to people. Like, you know how hard it is to get through a lantana thicket. So animals like swamp wallabies and bandicoots will feel very safe in those kinds of areas, and then they emerge in the evenings to forage.
And it's is there any similarities or differences in the diet of those two species?
There is. Swamp wallabies are one of the few macropods that actually are more of a browser rather than a grazer. A lot of macropods, when you think of like eastern greys and some of the other wallabies, they eat a lot of grasses, whereas swamp wallabies will eat a whole variety of browse. They'll also eat fungi. Whereas bandicoots are similar in that they'll eat fungi, but they also eat a lot of invertebrates as well. That's why you often see little diggings from bandicoots.
So Meredith, you're the botanist, I guess, the
field botanist,
flora.
yes.
And so what sort of methodologies would you be using for your field surveys?
It depends what we're looking to find out. If we're looking for what plant species are there, I would use, it's a widely recognised method called a random meander search. And you walk through and you record every different plant you see. And you keep meandering around the place. You're trying to pick up all the different types of vegetation or areas, little, beside rocks, under trees.
Are you off track?
Oh, absolutely.
Yes.
Absolutely,
Yeah.
yep. You're not going to see it all. You're going to see half of it and nothing of the really interesting stuff if you're on track. So the random meander is really good for finding out what's there. The next question might be how much of it is there. And that's when we'd use either a quadrat style assessment where you're looking at percent cover, or possibly a transect. A transect's good for looking at a larger area and a little bit more sort of broad brush. How big are the trees and how many trees are there, and have we got grasses or ferns or shrubs?
Okay. And when you're planning to do these surveys, does seasonality come into it?
Absolutely. Absolutely. It's much easier to identify a plant if it's in flower or if it's got fruit, or both, preferably. If you can get both, it's even better. I carry this around with me. It means I don't have to remember every detail about every different species, and it's really good for differentiating stuff in the field. There are plenty of other books. If I can't identify it using either existing knowledge or this, I take photos. I might take a specimen. We've got a license, so I can pick.
Okay.
And then I'll take them home and I'll go through books, and if I still can't work it out, I might actually post it online and say, guys, can you tell me what this is?
Okay. So my memory of this one is that it's actually got a dichotomous key at the start, but it's the field book then pretty much just gets down to the families and the genuses and their pictorial bits.
Yeah. This is a really good field guide because it says, look for this characteristic.
Yeah.
And this one looks similar, but they'll be different. It's definitely the best, and it's the Sydney Basin, so you're not being overwhelmed with all the rest of the plants that are out there, which is very easy. This one's pretty much just native, but a lot of them more common weeds. Okay. So the next thing that we might be looking for are threatened species, and the thing about threatened species is they're not very common. They often look really like something else, and you need to be able to say this is where they are, which is when we use the GPS. The animals live on the plants, and the plants live with the animals, and the way that they actually live together, and they've got all sorts of interactions and biotic feedback loops, and one without the other just quite simply won't function.
So Meredith, a moment ago you used an interesting term. You said biotic feedback loops in terms of plants and animals.
Yep.
What was that again?
A biotic feedback loop is, as an example, quick, give me an example.
Pollinators.
It's the way, pollinators is a good one. A lot of the orchids have very specific pollinators, so they produce scent and they develop an overall appearance that means they attract the specific, and often it's a wasp, and they will come, and they actually often think it's another wasp. They come over to do their little business and make more wasp babies, and what they're actually doing is picking up the pollen, and then they go to the next orchid with the pollen. And so without those wasps, that orchid won't be able to breed, so the orchids will die. But the wasps have got to get something out of it too, and what they get out of it is what...
Yeah, I know. Sometimes we debate that when we're looking for examples of mutualism.
Yes, absolutely.
Well, historically, there was a lot of small to medium-sized mammals, which we've now lost because of foxes, things like potoroos, that would turn over a lot of soil, like digging for invertebrates, digging for fungi, and all of those disappeared with the invasive animals that we brought with us, and so they're missing. So that's a missing kind of link. But animals that do that job, that biotubate the soils, are bandicoots, swamp wallabies to some degree, but even like the Australian brush turkey that's making a comeback in the Sydney Basin.
And lyrebirds.
Yeah, lyrebirds,
Yep.
yeah. And the action of turning over the soil, an important function of it is that they spread fungi spores through the ecosystem, which are really important for the health of the vegetation.
About 90% of Australian native plants have some kind of mycorrhizal relationship, which is where the root has an association with some sort of fungus. And that association relies on the fungi to actually bring nutrients to a zone where the roots can actually take it up. What is the question that we're trying to find an answer to, and will the data we're collecting give us an answer to that? Then there's the how are we actually collecting the data in the field, and there's a lot of established methods that can be used. A lot of them are described through Office of Environment and Heritage.
Yeah, there's guidelines for surveying specific species, and these are very well thought out, and a lot of research has gone into them to maximise your potential to detect that species, and also the best way to validate that data. And you need to know spatially where it is, so we always take GPS readings of threatened species, and mapping plays a big part in looking at fauna surveys and looking at the distribution of fauna throughout a particular area. I always try and validate the data by having more than one method confirm the presence of a species for fauna. It's just something that I like to do, and I know a lot of other ecologists like to do it as well. And it's also very good when you're trying to convey to the people that manage the land and the people that use reserves. It's always great to have a photo, so we always try and get a photo either through a remote camera or just physically taking a photo during the day of the species that are here.
Students doing biology in the HSC need to know a lot about the difference between handling and processing data versus analysing data. Do you guys think you could give us some examples of how on one hand you take care to handle and process your data in the field versus analysing it back at your base later on?
Sure. I mean, a lot of the processing also takes place indoors. Like, I will collect the data in the field, like that might be cards from the cameras or cards from the bat detector, and I take them back to the office to process. And you download all the raw data and you go through it, and you can end up with a large species inventory if you're lucky enough to be in a nice area. But that's not your job. The inventory is only part of it. You may be repeating a survey that was done 10 years ago, and so you're looking at change over time. So you need to analyse the results in the context of what it was like 10 years ago and what has changed, so a turnover of species. You also have to analyse other factors that might be influencing fauna. If there's a drought, for example, you'll get a different suite of species, or if there's a drought out west, some of the western species will move into the Sydney Basin. So just looking at, I got this number of species this year and I got this number of species this year, isn't particularly helpful with fauna, and it's probably the same with plants.
Similar, yeah. Quite similar. There's a number of underlying mechanisms that will drive a turnover of species. So that, for example, comparing a survey from now from a survey 10 years ago, I can look at what might have caused the change. But before you can do any real analysis of your data needs to be valid. And that's the process of going through and cleaning it and making sure it's basically formatted all the same. You've got the same, really simple things like the same units of measurement. Then once you've got your data set in order, you can start doing some analysis. And the first part of that analysis is usually descriptive stats. What a set of descriptive stats will do is give you a better understanding of what you've actually got. The descriptive stats says, yes, it looks like there are more of these than there are of those. But just because it looks like it from, say, a bar on a simple bar chart, it doesn't necessarily mean that it is. And that's where you start doing your more rigorous statistical analysis. And there are a number of different processes that you can use to do that, and a lot of that really depends on what you're trying to find out. I think the biggest issue for all of these different little bushland reserves around the Sydney Basin is fragmentation. Plants become separated from other plants in the same species, and it's the same for animals. They need to be able to migrate from one reserve to another to ensure you've got a maximum genetic variability. So given that fragmentation is probably, predation from feral species and invasion by weeds, they're really, really big problems too. But fragmentation is one that we could possibly do something about by educating people that live near reserves and educating people that live perhaps in a corridor between different reserves on how they can manage what they've got in their garden, so they're actually helping animals to move between.
Yeah. And in the reserves themselves, it's a fine balance between managing the flora and managing the fauna, because as we discussed before, swamp wallabies and bandicoots, for example, may be found predominantly in areas that have got very high density of weeds, because that provides the best cover. So you have to be very careful about removing weeds and making sure you're replacing those species with something similar, and that will take a long time. It's very hard for people to get their heads around 20 years to remove those weeds in that particular patch and then have it replaced in stages by similar vegetation. I think that's pretty tough for a lot of people to understand.
Yeah. Weeds are also really good for feeding resources.
Yeah,
and it's funny, I tend to think weeds for our native animals are a little bit like McDonald's. They taste really good, but they're actually not nutritionally that good for you.
What do you guys know about the Sugarloaf at Lane Cove National Park? How does it compare to other patches of remnant bushland around Sydney?
Oh, look, it's pretty nice in the context of where it is. There's some really nice reserves around the Lane Cove River, and there's a certain amount of continuity, even though there's some road crossings and things that have to happen for some species. Any areas of contiguous bush with any of the larger patches like Lane Cove National Park usually have a pretty good fauna assemblage, and the same with flora. It's a bit different if a reserve is completely isolated, then it's very, very tough for animals like swamp wallabies and bandicoots and sugar glides to persist there. But you have a constant influx of species into those smaller reserves, as long as they can make their way there safely, essentially. So it's pretty nice. It's a pretty nice bush for where it is.
So thanks for your time here, guys, in the bush. We'll let you get back to work doing your field surveys in a minute. Just a few real life questions. What's the most boring part of your job?
For me, it's processing the film that comes out of the cameras. There can be tens of thousands of photos of a lomandra waving a single leaf.
Lomandra being the strappy grass.
That's right.
Yeah, there's
Behind
one.
you there.
Yeah, a bit the same, processing field data, definitely.
And I guess maybe that comes into the next question, what drives you to keep going through those boring times and to keep going to do this job?
Oh, it's like a kid. It's like being a fauna ecologist. It's like a treasure hunt, like seriously. Like you can be looking through like 10,000 photos and then you see an echidna. And it might be the only thing you see in 15,000 photos. It's, oh my God, there's an echidna at the X reserve, It's awesome. Yeah. Like I just get so excited to see some of the species that have managed to persist, just so excited.
There's always something cool that you'll come across, something a little bit different, something that makes it interesting.
You're less excited than me.
I am less excited.
I was going to say, just what's the most random thing you guys have come across when you're out doing field work in Sydney bushland?
You sort of sometimes see in the news like there's some unusual exotic species that get around, like corn snakes and red-eared sliders. In the bushland just last week, I was walking through and a mother fox was coming down the trail, and she had her 3 cubs with her. And even though they're an invasive species and that's terrible, they're still a stunning animal. And she sort of saw me and she turned around very slowly and wandered off. And one of the cubs hadn't learnt that he was supposed to be scared of me, and he came and just sat and I had a look at him and he had a look at me, and lots of photos. So yeah, just that kind of stuff is awesome.
Would you recommend this job to young people, maybe your own kids?
Yeah, absolutely, absolutely. It doesn't pay great, but you get to be outdoors and you get to be involved in something that you can be really, really passionate about. It's a fantastic job.
Mm.
And you guys are your own bosses as well, so there's got to
Yes,
be something to be said for that.
Some days we've got the best boss and other days the worst.